Good morning, class. Welcome to online biology classes. As we are on chapter 1, the leaf, and already discussed various topics of the chapter. The previous topic we have discussed was vegetative propagation. Today's topic is artificial vegetative propagation. What is artificial vegetative propagation? It is the method of vegetative propagation which have been developed by man. So, it is a method of reproduction in plants or producing new plants by man. It is not a natural process, but this process is done by the help of man. There are 4 methods of artificial propagation. They are cutting, grafting, layering, and tissue culture. Now we will study each of these methods one by one. The first method is cutting. In this method, we take a part of stem from the parent plant. As you can see in the picture, a part of stem is cut from the parent plant. The parent plant is still rooted in the soil. The important point to keep in mind over here while cutting the stem is the stem should have axillary bud at the node. As you can see in the picture also, there are some growing branches on the stem. After you cut the part of the stem, place that part in the moist soil and leave it for few days. After few days, you will notice that the upper part of the stem has started growing as a shoot system, and the lower part of the stem will also develop roots. In this way, cutting method can be used to grow new plants. This method is used in sugarcane, rose, bougainvillea, and tamarind. Second method is grafting. This method is used to obtain a plant by combining 2 different plants. Now suppose if we want a plant of a new variety, we will follow this grafting method. In this method, we will take 2 plants of different kind. Example, we can take one plant of rose having flower of red color and one plant of rose having flower of white color. These are 2 different plants. Now what we are going to do, we are going to cut the stem from one plant and we are going to attach this stem on the another plant by tying it with a thread. So the part of stem which we have cut from one plant is called scion, and this part will be without roots, and this scion will be attached on another plant, and the part where the scion will be attached, that part will be called as stalk. And we will tie this scion with the stalk with the help of a thread. You can also see the same labelings in the given diagram. The stalk part should be having the root. After few days, scion and stalk fuse or merge and new cells develop having characteristics of both the original plants. This method is commonly seen in mango, rose, grapes and lemon. The example of rose plant which I gave, after the method of grafting, you can see that the stalk part of the plant or the second plant that we have taken on which we have tied the scion, it will have both the red flower as well as the white flower. So this is one feature of grafting. The third method is layering. In this method, a branch of a plant is pulled and its part is covered with moist soil, leaving the top part outside in the air. After some time, new roots develop from the buried part of branch in the soil. Now the branch is cut off from the parent plant and the part of the branch which has developed roots grows to become a new plant. As you can see in the picture, there is a plant. Now we have taken two branches of the same plant. One branch we have buried on the left side of the plant in the soil and another branch we have buried on the right side of the plant in the soil. Water both the parts left and right. After some time, you will see that the roots have started developing in both the right and left side of the buried part of the plant, and it develops into a new plant. So you can see this method is called layering, and commonly this layering is done in jasmine, lemon, China rose, etc. The last process or the method is tissue culture. Now this tissue culture is a scientific method of growing new plant. For this method, we require lab or laboratories. This kind of developing new plant, this kind of method is done in a test tube. So how it is done? Tissue culture involves growing of plants in test tube under laboratory condition. A small piece of plant is grown in a suitable medium in the test tube. Now what we do? We take a small portion of the plant in the test tube. And in the test tube, we have added a medium. What is a medium? It is a solution which help in the development of the plant. An unorganized mass of cell known as callus develops from it. After some time, we will see that callus or small plantlets or small group of cells have developed from that part of plant inside the test tube. The callus is put into a hormone containing medium which include cell differentiation and formation of plantlets. Now, after the callus develop in the test tube, we transfer this callus into another petri dish containing some hormone. What are hormone? They are the enzymes which help in the growth of the plant. After we transfer the callus into hormone solution, new small plantlet starts developing. The plantlets develop into an independent plant when transferred to the soil. Now, after the plantlets are developed, you can place the plantlets in the soil and each plantlet will start developing as an individual plant. A large number of plants can be produced from tiny piece of plant. Example of such plants are orchids. So that is all for today. Thank you, students. Do clear your queries, if any, in the comment section and revise the previous topics thoroughly.